Hello, how are you guys doing? I hope you're good. So excited to continue chapter 4, part 2. So we ended first part talking about the just general differences between the cell structure of gram-positive and gram-negative bacteria. Now we're going to just go a little deeper into that and look into that structure, talk about a typical cell wall, define plasmid, nucleoid, and endospores, and talk about the main structures of eukaryote and their functions. Actually, for this part, I would like you to do some more extensive homework than just watching my video, which is watch these three videos and understand how protein synthesis work, what is DNA replication, how is the DNA structure, and with this, you would be very close to understanding the eukaryotic cell organelles, because a lot of the organelles present in eukaryotic cells are have their functions related to protein synthesis and DNA replication. So I would like you to look into that. Look at the main structures and organelles of the eukaryote cells in your book. And then, so this last part here, I'm gonna let you do the work, okay? So let's move on to continue our talk about cell wall. So we're saying that cell wall can be different between gram-positive and gram-negative bacteria, right? And also, we mentioned how you have those that I call candy canes, which is intercalation of NAG and NAM, the two different sugars that make the sugar part of the cell wall, and that these candy canes are connected by peptide cross bridges, and that makes like a mesh structure that is going to be called peptidoglycan. Okay, so that's part of what makes the cell wall. And then I told you that this peptidoglycan layer is thicker on gram-positive cells and thinner on gram-negative cells. So what else is different between gram-positive and gram-negative? This is a gram-positive cell wall. So you see here that you have the gram. Let me get a pen here to tell you exactly what I want you to pay attention at. So give me a second. So you have here, connect the pen again. There we go. So we have here the peptidoglycan layer, right? And this layer is made by multiple layers of this sugar, right? This sugar and peptide combination in the gram-positive cell wall. So this is gram-positive. Other than that, you see this here? These are lipoteichoic acid. These are negative charged molecules, so negative charge that allows for ions that have a positive charge to flow in and out of this membrane. Okay, so it just helps the flow of ions in this cell wall. What do you have here? This is the plasma membrane. Okay, plasma membrane. You see, with proteins embedded on it, the phospholipid bilayer, just like in our cells, right? That's pretty much what I want to tell you. The difference between lipoteichoic and wall-teichoic acid is not much. One has lipid, the other doesn't, but they both have this function of attracting positive charged ions here. And then, now let's compare that to what is the negative. It's right here, gram-negative cell wall. So if you look at this one, look at that. You have the same plasma membrane. Let me get my pen again. There we go. The same plasma membrane, one layer, not multiple layers, of peptidoglycan. You see the candy canes here? And then right on top of that, you have another layer that is part, so all this is cell wall, right? This is plasma membrane, and then on top of it, you have cell wall. And all this is also made of an outer membrane. What is that outer membrane? That is similar to the plasma membrane. The difference here is that you have these molecules, you see these purple molecules here with this little tail, and these are called LPS. See, one of this is represented here, and that's the LPS. What is LPS? Lipopolysaccharide. Lipopolysaccharide is composed of an O polysaccharide and lipid A. What is important here? That the lipid A is the part that when these bacteria are multiplying and they're expanding their cell wall to be able to grow and then split in two, these LPS molecules get lost in the environment. When the bacteria dies, also that cell wall breaks down. So again, these LPS get released into the environment. And if the environment is your body, because the bacterium is inside your body, LPS causes, among other things, fever. LPS is a pyrogen, so don't forget that LPS causes fever, so it's considered a pyrogen. Okay? So that's the main function of LPS. LPS is also considered an endotoxin. We're going to talk about that later. So do only gram-negative cell walls have LPS? Yes. You're not going to see LPS in gram-positive cell walls. Okay? So that's one of the main differences, because this is a molecule that ends up being very, very virulent in gram-negative cell walls. Another thing I want to mention, when we apply the gram stain in the cells, why the gram-positive that you're seeing in the lab, right? The gram-positive cell walls don't allow the crystal violet to escape from inside the cell, while in the gram-negative cell wall, you see that leak of crystal violet when you apply the decolorization step, which is the alcohol. The alcohol makes holes on these cell walls. So these holes are going to allow, so this is the intracellular, right? Intracellular. So the dye that is inside the cell is going to be able to escape, and the cell is going to become transparent again. And then you do that counter stain with safranin. That's why the gram-negative cells always look pink after gram stain. And that's why the gram-positive cells, because they keep that crystal violet, are going to be purple. So let me show you a picture of that so you really know what it looks like. So you see, they keep the purple. So this purple in the gram-positive is actually, crystal violet. While the gram-negative, as they lost their crystal violet because the alcohol that is the next step after the crystal violet made poke holes on their outer membrane and on their plasma membrane, they are going to be counterstained with our second dye, which is, so this red comes from safranin. And you're going to be like, but why if you apply the counterstain to both, the gram-positive don't also get like red? They do, but purple is stronger color than red, so you don't really see the red from the safranin because that purple is more evident. Okay, so you don't visualize the purple, the red, because the red is sometimes you see as a light pink. Sometimes it's more red, sometimes it's light pink. So just general here statements about the difference between the two. Gram-positive cell walls produce exotoxin, so they produce toxins that are released in the environment, while gram-negative can also, other than exotoxin, produce endotoxin. Usually gram-positive cell walls are highly susceptible to penicillin because remember, we're going to learn here, and you should remember from anything you read, penicillin is antimicrobial drug that acts on inhibits the synthesis of the cell wall. So because it acts on the peptidoglycan layer, who is going to be more affected? A cell that has a lot of peptidoglycan to protect that cell, or one that doesn't rely so much on peptidoglycan because it has that outer layer of phospholipids? Of course, the gram-positive is going to be more affected. So penicillin ends up disrupting more the plasma membrane of gram-positive cells. Okay, and also disrupted by lysozyme, which are present, for example, in our tears, in our saliva. So lysozymes can break down bacteria that are gram-positive with more efficiency than the gram-negatives. Atypical cell walls. So here we're gonna have Mycobacterium and Nocardia. They have, so if this is a Mycobacterium, their cell wall here is made of something that is called mycolic acid. Okay, it's a waxy lipid, and mycolic acid is bound to their peptidoglycan. Mycobacterium and Nocardia are gram-positive, so I'm gonna write here that these both bacteria are gram-positive, but it's really hard to determine their gram stain because they have this waxy lipid around it. So the gram stain doesn't really work well, doesn't really stain this bacteria. So you need to do a different stain that you use instead of decolorization with alcohol, decolorization with alcohol and acid, a mixture of both. And that mixture is going to poke holes on this waxy lipid and stain the cell. We usually use carbofuchsin to do this acid-fast staining, and carbofuchsin will stain the positive acid-fast cells that have this atypical cell wall. Carbofuchsin is red. Oops. So when you look at their colorization, anything that you see in red here is probably Mycobacterium or Nocardia. Or if you want to be safer, you can say, you see, the picture tells me that this is Mycobacterium tuberculosis. But if you don't know, you can just say that these are acid fast bacteria. Okay? Cool. What else I want to tell you? So we talked about also remember that talking about atypical cell walls, mycoplasma, they don't have an atypical cell wall because they actually do not have a cell wall. They have some sterile molecules around the plasma membrane that helps for protection because remember they also don't have a skin, but they do not have peptidoglycan or anything like that. Mycoplasma are really tiny bacteria that a lot of times contaminate primary cultures in labs because they pass through filters. We're going to learn about how to sterilize by filtration. So then you think about mycoplasma. They're so tiny that they pass through the meshes of the filter, so you can really sterilize the medium if there is mycoplasma present. Sometimes, depending on, they can cause like lung infections that are confused with viral pneumonia because they're so tiny. They're hard to detect. Archaea, remember archaea either don't have a cell wall or have a cell wall of a molecule that is similar to peptidoglycan, but it's called pseudomurine. Okay, so they lack none and they have some amino acids that are different from the ones you see in the peptidoglycan. So that's archaea. And remember, that's one thing that distinguishes archaea from bacteria. Okay, so we also want to talk about plasmids. So bacteria have inclusions. I didn't really put that in the learning objectives. Just so you know, they don't have organelles, but they can have some little vesicles with different things that can be related to flotation or to energy reserve, okay? Bacteria can form endospores also. That's another property of some bacteria. Bacteria, usually we learn the bacteria from the genus Bacillus or from the genus Clostridium can form endospores. Endospores are resistant to harsh environmental conditions like desiccation, heat, chemicals, radiation. So anything that would make the bacterium die, the bacterium in those conditions, which are usually in the environment, not inside the host, because the host is warm, wet, proper pH, they usually don't have to form endospores inside host. But if Bacillus or Clostridium are in the environment, and the environment for some reason is too cold, too hot, something that can affect the bacterium, they have this tendency of reducing their cellular content. This is what happens. You see, that's the whole bacterium, but then they reduce to only this. So they just pack what's important here, and they form by a two membrane that is formed from the plasma membrane, a spore. So this is the endospore. And then the endospore is ejected from the cell. And this cell can go ahead and die because everything you need is here. These endospores can survive. Let me show you an electron microscopy of an endospore. So this is an endospore here. You see, it has this thick coat here. And this coat is responsible for protecting the material, the genetic material, and all the ribosome, whatever was kept into that little cluster. They can survive tens of hundreds of years in the environment. And then one day, when conditions are good again, they can go back to being a bacterium again. Okay, so the bacterium that generates the endospore is called vegetative, and once that happens, they can die, like I said. If that endospore comes back to being a bacterium again, that's a situation that, if I want to use an example to you, say, where does this happen? If you think about the permafrost that you see in the Antarctica, usually you have Bacillus anthracis there. So it's a species of Bacillus that is able to form an endospore in a very frozen soil. If that bacteria gets in contact with a human body that is much warmer and proper environment for them to survive, you can contract anthrax, right, which is the name of the disease. So that's one example. Another example would be canned foods. So when you have Clostridium likes is usually able to contaminate food that are going to be canned. If you don't add nitrite to this food, what's going to happen is that the Clostridium in that canned anaerobic environment, Clostridiums are anaerobe, so they thrive in the absence of oxygen. They will form an endospore, and that food was not properly canned because you didn't add nitrite. Those endospores, once you ingest that food, they will cause you some kind of infection. For example, botulism. Clostridium botulism is the one that can cause that type of condition. So with that, I think we're done with this chapter. Remember to go back and study the eukaryotic cell, protein synthesis in eukaryotes, and DNA replication. And I'll see you for chapter 5 lecture. Bye-bye.